once we know what the function of the protein is, that really only gives us a starting point into studies of structure and function relationships. Now, of course, this on its own is very important because that tells us an important characteristic of the enzyme that might tell us what the function of this protein is in the cell, or what's known as the physiological function. But if we're trying to figure out what is special about the structure of the protein that allows for this reaction to occur at a much faster speed than it normally would, uncatalyzed, then we need to know more about what are called intermediates within the reaction. And so note, we know what the reactants and the products are from the balanced equation, but within the enzyme there are other intermediate species such as substrate binding to form a enzyme substrate complex, reaction with another co-substrate in order to form what's called an intermediate, and then other reactions that are along the cycle that aren't necessarily inherent, or that there are no clues to within just the balanced equation. So this is really just a starting point. And so, like I said, things that aren't in the balanced equation may be really critical towards discovering what is important about this enzyme that helps speed up this reaction. So for instance, F and I, which are intermediate species along this reaction pathway, aren't in the balanced equation at all. And so this is not going to tell us anything about this. And so what we really need to know is we need to know time-dependent data of formation of all these, because these happen in sequence. So first, from the scheme, the enzyme, the substrate A binds to the enzyme to form enzyme substrate complex. This reacts with the co-substrate B to form this intermediate, and then another molecule of A reacts with this to form the enzyme product complex. So you see D and C are both bound to this, and then the products get released to regenerate the enzyme in its starting position, and then that can go around the cycle again. But as I stated, this happens in sequence, so there's a time dependence to this. So there's a cause and effect. And so when we have a cause and effect for each of these, then what we need to know is, when do these species form? Can we see them? And can we figure out in what sequence they appear? So for that, we need some sort of time-dependent plot where x-axis is time in some unit. Typically we use seconds or minutes, but I'll put seconds here. And then, depending on what we're looking at, we need to know the concentration of a particular species. Starting out for any enzyme study, as I mentioned in the last video, what would be nice is if we can find the time dependence of product formation. So depending on how we're able to detect this product, so if we're able to detect this, for instance, by UV vis absorption spectroscopy, then what we can do is that we can have a cuvette, as you probably used in your general chemistry classes or organic chemistry, and we can make a solution of the protein. We'll just pretend like the protein's red, just to make it easy on ourselves, okay? And then we add A and B in here, our substrates, and then if we can see product, if we can see the product C, for instance, after we add this up, we'll see an increase in the product C. So we'll color this blue, and we'll get some plot that at 0, at the initial time, there will be no product, and then we'll see the rise of it as time increases this way. Okay? And so this gives us an idea. This is what's called, in this case, it's called a continuous assay. What's nice about continuous assays, you can get a lot of data points, because in this case, we don't have to do anything except for shine light, so UV or visible light through, and then detect the absorbance. This is the detector. and then we can determine the concentration of the product. As we discussed before, for protein purification, we can detect this by Beer's law. And what's nice about this method is that we just follow the formation of product while it's in solution using the UV-vis absorption spectrometer. And in this case, we don't ever have to do anything to the protein. In other words, we don't have to denature the protein. We can see the product by UV-vis absorption spectroscopy. We don't need any extra sample prep. We can do everything inside of the cuvette. And so this is what's called a continuous assay. And we may not get a perfect line like this, but we may get a larger number of data points, a very large number of data points, which makes it look almost like a continuous trace. So this is what we call a continuous assay. Now, if we can't detect the product, for instance, by something that we can do a continuous assay with, such as by UV-vis absorption spectroscopy, we can do what's called a non-continuous assay. And this, we may have a situation where we have to stop part of the reaction. For instance, if we have a test tube containing our enzyme, and A and B in it, and we let this go for some period of time. Now, if we can't detect this product directly, what we may need to do is we may need to take out an aliquot of it. So we lose some of this volume, and we add a small amount of it, maybe to another test tube, and say we can't detect C directly. So we have to do, for instance, maybe a derivatization reaction, or C, we do some sort of side reaction, we can convert to some product X, which we can then detect by UV-vis. Okay? And so what this does is this stops the assay. So this stops the assay for this little aliquot here. And that's why we call this a non-continuous assay. And so we have to do some sort of sample prep. So we take this, we add our reaction, and then this turns into some other color that we can detect by UV-vis, which is related to the product C. Okay? The problem with this is, unlike a continuous assay, which has many, many data points that is only limited by how quickly, for instance, our spectrometer can collect, we're going to have far fewer data points on this. So for instance, for a time course, for C using a non-continuous assay, you might expect something that looks like this, where at 0, 0 we have a point, and we can collect points at different intervals, but we're not going to have quite as much data as we would for a continuous assay. For instance, in the case of using UV-vis absorption spectroscopy. And so these are two very basic different types of assays. There are other types that we're not going to get into that are beyond the scope of this, but these, it's important to think about when you're developing an assay. One, what can you detect? Importantly, can you detect out of these products? And if you can you detect them by a continuous assay, which is going to give you more data, and so you're going to get more reliable information out of that, or do you have to use some sort of non-continuous assay? And while you'll still get data, it may be less reliable. And in that way, you may not be able to get quite as good data, but you may be able to get enough data to start the study at least. But importantly, what's important is that we need time dependence of product formation before we can look at different things, such as formation of intermediates, which we can then use to determine structure-function relationships.